HIV infects cells via two surface molecules. CD4 is the primary receptor for the virus, while the chemokine receptors CCR5 and CXCR4 act as co-receptors for the viral infection of dendritic cells and naive T cells, respectively. HIV binds initially to CD4 via the envelope glycoprotein GP120. Interactions of the virus with the CD4 and the co-receptor allow virus uncoating and the entry of the nucleocapsid containing the viral genome into the cell. The viral reverse transcriptase, which is an integral part of the viral particle, copies the RNA genome of HIV into double-stranded DNA. The viral integrase then mediates the integration of the viral DNA into the chromosomal DNA of the host cell. In this state, the virus is latent. That is, it can persist in the cell in an inactive state. Reactivation of the virus occurs when the host cell becomes activated and viral transcription is initiated. This results in the accumulation of viral proteins, as well as genome-length RNA transcripts of the virus. Viral particles assemble at the cell membrane with copies of the RNA genome and bud off to create a new viral particle. Maturation of this new virus particle continues after it is budded off from the host cell to create a new infectious virion with its characteristic nucleocapsid morphology.